anatomy of hardwood tree species. So if you can share your PowerPoint with us.
Good evening, everyone. Thanks a lot for providing me this opportunity on the international platform that is Global Plant Science Virtual 2020. I'm here to present my novel work on a topic that is wood anatomy of hardwood tree species. So this is a field where unexpectedly I joined during my PhD days, and I got to explore more of it. And I'm here to share my work, which was based on the hardwood tree species. Wood, as we all know, is technically the secondary xylem of the mature shrubs, trees, and lianas. And in any vascular plant, it is externally covered by the bark, which is the secondary phloem. This is a result of secondary growth produced by the cambium tissues in the case of plants. As far as the wood anatomy is concerned, it is a highly conservative feature. When I say conservative feature, it means that all the elements or the wood elements of the plant, they show rarely any changes in its structure to a large extent under extreme environmental conditions. And that's the reason when you take up wood anatomy as a criteria, it becomes a very important criteria for authentic identification of plant species. This field has got quite a lot of applications, and as we all know, wood finds various uses even in our daily life, and so it gives a very good scope for research, like in the field of taxonomy, plant ecology, archaeology, etc. There is a specialized branch under this which is called as fossil woods as well. And when we talk about the wood, the first thing that comes to our mind are the growth rings or the annual rings. These rings are quite easily visible when a cross section of the log is taken, and you can observe these rings in the form of concentric circles right from the center to the periphery of the wood. When you say annual ring, it indicates that one ring represents one full year of growth. Each growth ring corresponds to one early wood and one late wood. Early wood corresponds to the summer or the spring wood, which is considered to be a favorable season for the growth of plants. Similarly, the late wood comes under autumn wood or the winter wood. Autumn wood or the winter wood, which is considered to be an unfavorable season. That is the reason one growth ring or the early wood and the late wood becomes very distinct in the case of species which are deciduous generally. The reason is they grow in certain conditions which are where the climatic conditions are like the seasons are very distinct, and the distinctness in the season is reflected in the growth rings as well. And this field is known as dendrochronology, where we are aware that the counting of the growth rings right from the center to the periphery can reveal the age of a tree. But that is not the only thing that we can find out from the growth rings. Growth rings can reveal quite a lot of information even about other features like the stress conditions faced during a certain year, or there can be changes in the growth rings based on the availability of water to the plant. And growth rings are somewhat distinct even in the case of fossil wood. So it gives an idea about the phylogeny of extinct plants and various such fields. So this becomes an important feature in the case of wood study. When you talk about wood, broadly speaking, there are two types. The first type, the types of wood are based on the type of species from which the wood is obtained. Like the first type is called as softwood, and this is obtained from the gymnosperms. These are not the flowering plants, but belongs to phanerogams and comes under one of the higher group of plants. And we have conifers and all, which are good softwoods, and they are very good as timbers as well. Similarly, the other type is called hardwood, and this is obtained from angiosperms, the true flowering plant. In India, we have both the types of wood available, and more of them are available as hardwoods, and we have great diversity of them in our country, in India. If you see the anatomical structure of these two types of wood as a true block, this figure, as you can see, is a true block. That means it has been cut in a section where you can visualize the wood in three different angles. The upper part is called as the cross section or the tier, and then we have two longitudinal sections. One of them is the radial longitudinal section, and the other one is the tangential longitudinal section. In the case of softwood and hardwood, all the three types of sections show slightly different features. The distinct features between both of them, if you see, the markedly distinct feature is the presence and the absence of xylem vessels. In the case of softwoods, the vessels are absent. Instead, they have more number of tracheids within them. In the case of hardwood, there are vessels present. Tracheids are relatively less. Because of the presence of these vessel pores, the hardwoods are also known as porous woods, and the softwoods are also called as non-porous woods. Here also, as an example, I can tell you how the wood feature can reveal an example to say that the angiosperms have evolved from the gymnosperms. Like for example, in the case of softwood, tracheids carry out dual function of conduction as well as mechanical support. In the case of angiosperms or the hardwood, these two functions are performed by two different tissues. Vessels perform the function of conduction, and fibers, which are abundant, perform the function of mechanical strength. So this, as an example, I can tell you, study of the wood can reveal a good data of information about the phylogeny and evolutionary aspects. So with this background, I'll come to my study, which was based on the hardwoods. When I joined for my PhD at the Forest Research Institute at Dehradun, my idea was to study the effect of wood anatomy on the effect of environmental stress conditions on the hardwood trees. I wanted to find out what kind of stress the plant reflects in its wood anatomical features due to some anthropogenic activities, and I narrowed down to mining conditions. In India, in the eastern part, there is the Chota Nagpur plateau, which is known for a good number of mines and which are quite old as well. So I shortlisted the oldest iron ore mines in the state of Orissa in India, where there are quite a lot of forests which are present. The forests belong to the moist deciduous type, and there are quite a lot of huge trees which are standing and exposed to the mining conditions since a long time. So I found this could be a very good area for study, and I can easily collect the wood samples, and I can study them by comparing the stressed wood samples with the controls of the same species. So my study area is in this Orissa state in the southeastern region in India, and in Orissa I selected these three iron ore mines. These three are very old iron ore mines, and open cast mining has been carried out since many years and decades actually. So to give an idea about the conditions, these are the photographs of the mine. As you can see, a lot of excavation activities are being done, and because of the factories located out there, pollution levels are also relatively high. And along the mines nearby, there are forests also which are available, because of which, after carrying out this survey, I selected this as the best area for my study. So the way I proceeded was, first of all, the sampling procedure. Now, because I had to collect samples from trees from a huge forest area, so I had to make sure that I cover almost the entire area and cover the maximum number of species to give an exhaustive good result. And for that, I followed a systematic random sampling method. So as an example, I'll show you here. This is one of the mines of my study, and this is the core area of the mine. And from the core area, what I did, because the forest was surrounding in all the directions, I selected three different lines across the entire area, which was, that means from the core area of the mines to the fringes. And in each line, I laid at an approximate equal distance, there were three quadrates which were laid. And within the quadrant, whatever tree species were comparatively accessible, considering all the practical aspects, I collected wood samples from the tree. This I did for the three lines in one mine and for the other two mines as well. So coming to the collection part, it was comparatively a physically demanding task, but it was worth it. The instrument which I used was increment borer. Now, using this instrument, I had to approach the tree, and at the DBH level, I had to make sure that the outer small portion of the bark is removed. And then when I reached the wood, I had to insert this borer, and the amount of strength required obviously depended from one species to the other. But after inserting the borer, I had to twist it a little bit, and when I take out the borer, I get the wood disc like this. So I collected as many discs as possible, but I had to ensure one more thing, that I cause least damage to the tree as well. So these discs were collected, appropriately labeled, and for every individual tree from which I collected the sample, I recorded the other preliminary data as well, even the GPS coordinates, the girth, the circumference, the height approximate, etc. So like this, from the study areas, I collected samples from about 90 trees in the mined size, which belong to 22 species across 16 families. And because this study had to be done for understanding the effect of this mining stress on the wood features, so I had to do a comparative study for which I needed control samples. Now, in this map of India, Orissa is over here, and I had to take control samples from an area which does not have mining or does not face mining stress, and overall it faces negligible stress as well. So the one of the areas I selected was the neighboring state of Jharkhand. In Jharkhand, there is a Dhanbad district where there is a good forest cover. So I selected that area where, after a survey, I found that most of my species were also available for effective comparison. So in that forest area also, I collected samples in the same manner. And from the Dhanbad district, Jharkhand, I collected from 24 trees belonging to 10 species. There were some species which were not available, and for that I had another good option, and that was from my institute where I did my PhD, that is FRI Dehradun in Uttarakhand. There is a xylarium which is located. A xylarium is a place where there is a collection of wood blocks which has been collected and preserved over many years. And in the xylarium of FRI Dehradun, there are more than 25,000 species from which the wood samples are kept and still preserved. So this becomes a very good spot for my collection, and I got all the other species for my effective comparison as controls as well. So this was one of the locations at FRI, and the other control samples from the Dhanbad district of Jharkhand. So in that way, totally I could get 96 control samples. And here also, for every wood sample from the xylarium, there is a respective accession number available, which was noted down appropriately, and the samples were selected in such a way that they belong to as much nearby area of the study area as possible. Though the sample is available over here. And after the control samples were collected, all the samples were ready. The next part was to authentically identify. And that means after authentically identifying, the control samples were collected. And these were the 22 species from which the samples were collected. These 22 tree species were authentically identified at the Botanical Survey of India in Dehradun. And then it was finalized. The next part, after getting the wood samples, the next part was carrying out the sectioning part across all the 3 angles, followed by preparing the permanent slice and collecting the microscopic data. This sectioning, for the sectioning purpose, I required a microtome, that is the Leica sliding microtome, and using this microtome, which was available at the MS University Vadodara campus, I could get sections as thin as 8 to 20 microns. So this was carried out first, and those sections were subjected to standard laboratory procedure, using which permanent slides were prepared by using DPX as the mounting medium. There was one more thing which was carried out, and that was maceration using the Schultz method. This was required in order to estimate the vessel and the fiber characteristics across all the 3 angles. So like this, the samples and the sections were ready, and this was followed by collecting the microstructure data for all the sections of the stress samples as well as the control samples. For collecting this data, the reference which was used was the microstructure format that was given by the International Association of Wood Anatomy, that is IWA. For each wood sample, there are approximately 183 features that can be studied. Approximately for 2,000 subsections all the data was collected, they were recorded, and they were tabulated accordingly. Besides this, um, one more thought which came across my mind during the course of my research was that because the three species I selected and the study area which was selected was in the iron ore mines, and iron being an element, a metal element, so I could hypothesize that in the three species under study, it could be possible that the tree must have taken up the iron element from its roots and it could have been accumulated in the woody tissue. So I wanted to include this parameter as well by quantitatively estimating the amount of iron in ppb or ppm using the ICP-AES facility which was available at IIT Bombay. For that, what I did was using the wood chips, I first prepared an acid extract. Acid digestion was carried out, followed by filtration using Whatman filter paper number 42, and the acid extracts were prepared. These acid extracts were subjected to this instrument ICP-AES available at IIT Bombay, and I got the results at the ppm ppb level. Coming to my results of the study, the first thing I would like to show all of you is that my study was based on 22 different species, and the first major part of my study was based on the anatomical features of the wood, of the secondary element. So yeah, I'll show you some excerpts from the microstructure or the photomicrographs which are taken using the AxioVision software. And as you can see in some of the species, the features are quite remarkable as far as the quantitative features are concerned. Like in the case of this one, in Mangifera indica, C stands for the control sample, S for the stress sample. So one can see the difference in the vessel diameter in the cross section. And this difference seems to be, looks quite significant. Similarly, there are differences in other species as well. And like in the case of Dalbergia latifolia, in the TLS, we can see the difference in the ray dimension. This can be seen in the TLS. And then you can see in the macerated slide of Shoria robusta. Shoria robusta is one of the very good timber species, and fibers play a very important role in providing strength to the wood. So the fiber length showed a significant variation in the case of Shoria robusta. Then in the case of Cydium guajava, here these are the intervessel pits. These pits can be visualized under high power on the walls of the vessel, on the xylem vessel. And the size or the dimensions of these pits also plays a very important role, because these pits on the vessel walls form the opening through which the conduction of the sap can go from one vessel to the adjacent vessel as well. So in those stress conditions, these pits can show some plasticity. Similarly, in other species also, like Artocarpus heterophyllus, one can see a difference in the vessel length as seen in the TLS. Here we can see a longer vessel in the control, and in the stress sample, the vessel length have become relatively smaller. Then in the case of Tectona grandis, which is another very good timber in India, the fiber dimensions show a remarkable difference. Fibers, again, as I told you, becomes a very important feature for timber species. Then coming to Schlesiera oleosa, another native species, you have the ray dimensions have changed. The ray parenchyma actually in a wood is associated with storage as well as lateral conduction. So here what I want to highlight is that the plant under its own stress conditions, as an overall result, I can say that according to the requirement of the plant, the wood element can partition its energy by changing the dimensions of its quantitative or the main element. And this is reflected in the control and the stress samples. Similarly, in case of Fongamia pinnata, there is a difference in the vessel frequency which is quite apparent. So like this, there are 22 species for which I have carried out the analysis and I have tabulated, which I will show soon. And because during the course of the analysis, there were some qualitative features of the wood as well. Overall, for the qualitative features, there were not much significant variations, and that becomes another very important point to emphasize on the point which I told in the beginning, that wood anatomy is a very conservative feature, that the qualitative features of the wood generally do not change much. But under extreme environmental conditions, there can be a slight variation, which I saw in two of the species which are very prominent. So I thought of going for SEM at the Punjab University, Chandigarh, And upon doing that, one of the results which I came across, an interesting finding which I came across was that in the Ziziphus jujuba, in the stress sample, I could see the coalescence of the intervessel pit which was observed. Coalescence denotes the fusing of the adjacent intervessel pit. So sometimes the plant might require to increase the size of the pit for effective and more water conduction owing to drought conditions. So that was very nicely reflected in the Ziziphus jojoba under SEM. Similarly, in this is the photomicrograph, the electron micrograph of Tamarindus indica, a control sample. Again, this is the intervessel pit, and one can see the small opening on the intervessel pit in the control sample. In the same species, Tamarindus indica, in the stress sample, we can see that these pits have slightly, almost they have closed. This phenomenon is known as vesturing. And vesturing, though it is a taxonomically family feature, but in certain environmental conditions like stress, the plant has the ability to induce vesturing in the intervessel pit. And that is the reason. The reason is because when the vessels are conducting something which is slightly unfavorable for the plant, it might close the pit so that it doesn't go to the adjacent vessel, or it can be induced by something called cavitation as well. So this kind of qualitative features also I could find out as an interesting result. Then this table gives you a comparative analysis. When I recorded the entire quantitative data for all the 22 species for all the parameters, like these are the fiber parameters, the vessel parameters, and the ray parameters. Upon compiling them and analyzing them statistically using the student's t-test, I could find out one common conclusion, that there was no common trend across any of the species which was found. Like in the beginning, the hypothesis was that there will be some effect of mining, and the effect was seen, but without any consistent trend. Like in this table, as a concluding table, I have put over here that the red downward arrow indicates significant decrease in that dimension, in that parameter. And the green boxes with the upward arrow indicate significant increase in that parameter. The rest of the blank boxes indicate no significant variation. So as one can see over here, across all the 22 species, there is no common trend that is observed. And this points out to one very important fact, that the change of plasticity or the energy partitioning in the wood anatomical features depends on the species level. The plant has got that ability that according to the conditions that the plants are facing, they can change the wood anatomical quantitative features up to a certain extent, but not more than a limit. But qualitative features showed more or less throughout they were the same. And that's the reason it becomes a very important criteria for wood identification. Then the other parameter which I did was the estimation of the iron element content in ppm. So here I predicted that all the stressed samples of all the 22 species will have high iron element content as compared to the control counterparts. But to a surprise, I found that out of 22, 4 of the species, they show relatively less iron content in the stressed sample as compared to the control. There could be many reasons for this. Like wood elements like the xylem vessels, they show a phenomenon called as stylosis, where they are able to plug the vessel pores so that no uptake is possible or no conduction is possible across it. So might have been possible some species are able to evade the uptake of the iron element across its tissues, and they are able to conserve it better. So it depends from one species to the other, and more studies can be carried out in this way. And as a final conclusion, I would like to show this table where my entire study was based on 3 different parameters. The first one was the microstructure analysis, followed by this iron element content, and there was one physical test also which I did, was the estimation of the specific gravity. Specific gravity is associated with the density of the wood, and that also I took it as a physical parameter, because when you compare the vessels or the fibers or the parenchyma, the fibers, in more number of fibers lead to more dense wood, and more vessels and more parenchyma reduces the specific gravity. So according to the quantitative parameter changes, the specific gravity correlated were different. So I considered these three parameters for all the 22 species. And when I did the final conclusion, I could point out that this species, that is the Shorea robusta, showed relatively very high level of changes across all three parameters. And that's why this species, actually this Shorea robusta is the most native species available out there. This is commonly known as sal in India, and that the forest over there are known as the sal forest. So this species is very common out there, and it has been able to adapt itself to those conditions by modifying its quantitative elements to a certain extent, but still surviving in large numbers. Similarly, on the other side, if you see, Buchanania lanzan shows the least amount of changes across all three parameters. Again, this can be studied further, and from my study, what I could find out was that the implication which I want to highlight from my research is that in the mining areas, once the excavation and the mining has been completed, the restoration of the mines is carried out. For restoration, selection of the species should be correct enough. Instead of planting any random species or introducing exotic species and destroying the local ecosystem, it is always advisable to go for native species or subspecies which are relatively very sturdy and tolerant in those stress conditions. So from my study, I'm able to point out that these are the two species which are very suitable. And the another thing which one can remember is that for restoration or replanting in those mining conditions, the germplasm could also be collected from the local forest itself, because the adaptations and the sturdiness is inherited in the genes and that can be transmitted well, due to which the species will be able to survive better in those conditions. That is the main implication of my study. So for this, that's my final conclusion, and I would like to acknowledge first and foremost my guide, Dr. Sangeeta Gupta Madam, under whom I did this exhaustive work. And these are my parents who have always supported me throughout, and because of whom I could be able to reach this position. I would also like to highlight Dr. K.S. Rajput sir. He is an associate professor at the MS University Vadodara, and his lab was the one where I did the microtome structure and the slide preparation. She is my colleague Ankita Gupta, with whom I did my PhD together. She worked on the coal mines of Jharkhand. He is Mr. Raman Notial sir, statistician, and for this exhaustive work I required a lot of statistics, for which he guided a lot. And I would also like to acknowledge SIS College of Arts, Science and Commerce, who during the end of my PhD days, they were very supportive for thesis making and compilation, and this is the place where I'm working at present as an assistant professor in the botany department. Thanks a lot, everyone.
Thank you very much. So I
Thank
at least half of those woods are in Jamaica. So my question to you, when are you coming to look at our woods?
Yes, I'll be coming soon. I'll be coming soon, ma'am. Thanks a lot.
Thank you. Okay. Take your screen off. We'll have next Shukla from JK Paper Limited.